So I'm excited to have the opportunity today to present our research on the genetics of Chiari Type I Malformation. So the outline for my talk is to discuss the general motivation for studying the genetics of a disease, walk through several lines of evidence that suggest a genetic component for Chiari, talk about some of the challenges that we face when studying Chiari, particularly with respect to the genetics, present our current research approaches and results, and then discuss some of our future work. So why study the genetics of a disease? Well, if we identify a gene or genes that increase susceptibility for a disease, this could lead to the development of potential genetic tests. And this could be especially useful for diseases such as Chiari, where symptoms are often vague and nonspecific, which can lead to lengthy and often misdiagnoses. In addition, if we start to better understand the potential genes that are involved in disease development, this could lead to a better overall understanding of the underlying disease mechanism and what specific biological processes play a role. And finally, there's the possibility to develop new therapies and treatments down the road that maybe target specific biological pathways that are misregulated during development. There are several lines of evidence that suggest a genetic component for Chiari. These include familial clustering, which refers to the identification of families where multiple members are diagnosed with a disorder. But I should point out that just because we observe a disorder runs in families does not mean it's necessarily genetic. So it could be due to chance, the environment, or genetics, which is really why we look to see if there are additional pieces of evidence also suggest a genetic component. In addition to familial clustering, we also see evidence from twin studies where we see a higher disease concordance between monozygotic twins compared to dizygotic twins. And we also see co-segregation with known genetic syndromes. So the idea here is that perhaps there's an underlying genetic link between the diseases. An additional piece of evidence stems from a family study which estimated heritability of various posterior fossa measurements. So heritability is just referring to what proportion of phenotypic variation is attributable to genetic variation. So if we identify that something is heritable, it's likely to have a genetic component. And this is especially important since the posterior fossa is thought to play an important role in disease development. And finally, the first linkage screen for Chiari was published back in 2006 by our group, and we identified 2 regions of the genome that showed evidence for linkage. So now I'd like to switch gears and talk a little bit about some of the challenges that we face when studying Chiari. And really the primary challenge that we faced is disease heterogeneity. And by disease heterogeneity, I'm referring to both clinical variability as well as potential genetic variability. So if you look at this sort of simplistic diagram, each circle corresponds to a patient, and each color reflects differences in clinical presentation. So we know that although patients may be diagnosed with Chiari Type I Malformation, that a lot of variation exists in terms of symptom presentation, age of disease onset, degree of tonsillar herniation, surgical outcome, as well as the presence of associated disorders such as syringomyelia. So where this variation is particularly problematic as it's poorly understood and may in fact be related to variation in terms of different genetic and environmental factors that could contribute to susceptibility for disease. So in this particular diagram, I'm showing 2 disease genes, G1 and G2, and a main environmental factor, E1. So our working hypothesis, and really the model that we've based much of our current research approaches on, is as follows. So it's very possible that there are several major genetic and or environmental factors that increase susceptibility for disease. And that would be shown here as the major red, orange, and gray. But within each of these groups, there's likely still additional variability, and that could be influenced by other genetic and or environmental factors. So our primary goal is to first try to identify some of these major genetic factors, and then once we do that, we can start looking at this additional variation because there might be some genetic or environmental modifiers for disease. So when designing genetic screens, etiologic heterogeneity becomes really important to consider. So if we take sort of this traditional approach and ascertain a large collection of Chiari patients, as shown here, and try to identify genetic factors, we're likely to have very low power or ability to detect even a single disease gene due to the potential heterogeneity. So one way we can try to approach this is to start to address this potential genetic heterogeneity and identify more homogeneous groups of patients and our families, and then look for genetic factors within each of these groups. So for our current work that I'll be presenting for the remainder of the talk, we've done both this sort of traditional approach, so ascertaining a large collection of patients, and then we've also taken alternative approaches that begin to address some of this heterogeneity. So the results I'll be presenting come from variations of a whole genome linkage screen that we completed, where the main goal is to identify regions of the genome that are segregating with disease in families, and then look within these particular regions for potential candidate genes. So currently, we're ascertaining Chiari Type I Malformation families. We are including families that contain at least 2 sampled individuals affected with Chiari with or without syringomyelia. And the Chiari diagnosis is based on either patient report, medical record, or MRI. And if we have MRI data available, we are classifying individuals as affected if both tonsils are herniated 3 millimeters or more, or one tonsil is herniated 5 millimeters or more. We're also excluding families where someone has been identified with a known genetic syndrome that has been previously reported to be related to Chiari. And then we're also excluding individuals thought to have acquired forms of Chiari, such as due to a brain tumor. We're collecting cranial and spinal presurgical MRIs, as well as questionnaire data, which encompasses things such as symptom presentation, presence of associated conditions, age of disease onset, pregnancy history, which starts to get a bit more at potential environmental factors, as well as surgical outcome, which will mainly be assessed based on symptom resolution. So for our initial analysis, we really took more of a traditional approach. So after data quality controls and cleaning, we obtained over 500,000 genotypes on 367 individuals from 66 multiplex families. And for this particular analysis, because the underlying genetic model for Chiari is unknown, we performed both a parametric and a non-parametric analysis. So the main difference between these two types of approaches is the need to specify a genetic model. So for the parametric analysis, we're assuming an autosomal dominant mode of inheritance, which is fairly consistent with our families. We're also assuming a rare disease allele frequency, and conducted an affected-only analysis, which just means that we're not going to lose power if we have unaffected family members that are misdiagnosed or actually carry the disease allele. For the non-parametric analysis, we performed 2 different types of models, and we also, for both, ran a 2-point and a multi-point analysis. A 2-point analysis is going to test for linkage between a single marker and the disease locus, and a multi-point analysis utilizes information across many markers when testing for linkage, and this can actually be more informative and can help to better localize susceptibility genes. So here I'm presenting a summary of our 2-point analyses across the 3 different models I just described. So on the x-axis are the chromosomes ordered 1 through 23, and each chromosome is shown in a different color. And on the y-axis is the LOD score, or logarithm of the odds, which is our way of assessing evidence for linkage. So traditionally, a LOD score exceeding 3 would be significant evidence for linkage, and a LOD score anywhere between 2 and 3 would be more suggestive evidence for linkage. And so what you can see is that we see multiple regions of the genome across the 3 models where we do have significant evidence for linkage. In fact, we see some consistency across models that are colored in red. So those are regions on chromosome 1, 4, and 8. But in some cases where we do see very significant LOD scores, we don't have a lot of support with neighboring markers, which is not quite ideal for a linkage analysis. So here I'm showing a summary of our multi-point analyses. Unfortunately, we did not obtain any maximum multi-point LOD scores, even exceeding 2. So I'm just presenting those regions of the genome with LOD scores exceeding 1.25. And those are regions on chromosome 2, 8, 2 regions on 9, 1 on 12, and 1 on 18. So at this point, we looked within these candidate intervals for potential genes, and while there are good biological candidates under each of these linkage peaks, given our minimal evidence for linkage at any one peak, we decided to take 2 additional approaches before pursuing these regions. So these alternative approaches, the hypothesis is that one of the reasons that we're obtaining really minimal evidence for linkage is due to this potential heterogeneity. So our goal is to increase our power by identifying more genetically homogeneous groups of families. And we'll use 2 approaches to do this. The first approach is more of a qualitative approach, where we're going to perform a stratified analysis using clinical criteria. And the second approach is more of a quantitative approach. So we're going to perform an ordered subset analysis, which is a type of stratification analysis, and we're going to use posterior fossa measurements. So for this first approach, the primary question is, well, what clinical criteria should we use to stratify our families? So over the years, there's been accumulating evidence supporting an association between hereditary disorders of connective tissue, such as EDS or Marfan syndrome, and Chiari malformation. So this has been published previously by Milleret and colleagues, and we've also noticed this in our own families, with an estimate of about 20% of the families that we're currently trying to enroll report that at least one of their family members has been diagnosed with a connective tissue disorder, and this is usually EDS. So although families such as these would be excluded from our genetic screen because they would be considered syndromic families, we've still noticed that many family members appear to exhibit CTD-associated symptoms and/or conditions. And while these symptoms and conditions are definitely not specific to connective tissue disorders, our thought was that perhaps these families differ clinically, or in maybe a small proportion of cases, we have some underdiagnoses of connective tissue disorders. So in either case, we thought, well, maybe these families actually have different disease etiologies. So we decided to analyze them separately with the hope that even though we're reducing our sample size, that we'll actually see increased evidence for linkage. So we took our 66 families and stratified based on a family history of a CTD-related symptom and/or condition, examples of which are shown here. And we were pretty lucky in that this gave us about a 50-50 split in our population. So for the CTD-positive group of families, that was 34 families, and the CTD-negative group of families was comprised of 32 Chiari families. We then reran our linkage analysis within each of these subsets. So here I'm showing the same summary of our 2-point analyses, but now performed in each of the family subsets separately. So the top panel shows the CTD-positive results, and the bottom panel shows the CTD-negative family results. And there's a couple important things to notice. One, even though we've reduced our sample size, we still see significant evidence for linkage to multiple regions of the genome. And in fact, in some of these cases, we see much more support in terms of neighboring markers that are also significant within the same region, which is really what we'd expect to see. In addition, we also see different regions of the genome that pop up in the different subsets of families. And this would also be consistent with the idea that perhaps different genetic factors may actually be involved in disease within these subgroups. So here's a summary of our multi-point analysis. And remember, when we analyzed all 66 families collectively, we did not obtain a maximum multi-point lot score even exceeding 2. But now we see, even with a reduced sample size, lot scores exceeding 2 across multiple regions of the genome, as well as exceeding 3 in one region of the genome shown here with chromosome 8. So our current focus has been on the CTD-negative group of families, and specifically we're focusing on regions of chromosome 8 and 12 due to potential biological candidate genes. So our candidate interval on chromosome 8 is this region here, boxed in red, 8q22.1. And while there are multiple candidate genes, the one of particular interest is this gene GDF6. So this is actually a very small gene. It's comprised of only 2 coding exons and a large 3' untranslated region. The growth differentiation factors are members of the bone morphogenetic protein subfamily of transforming growth factor beta signaling ligands, and they're known to play an important role during development. Now, interestingly, GDF6 mutations have been associated with a wide range of phenotypes. This includes ocular phenotypes as well as skeletal phenotypes, including Klippel-Feil syndrome. And this was really exciting for us to see because we know that Klippel-Feil syndrome has been associated with Chiari malformation, and it's been estimated that about 3 to 5% of Chiari patients have been diagnosed with Klippel-Feil. But what was even more encouraging is that when we started to look at other candidate regions, on chromosome 12, we noticed another GDF gene was present. So it's a very similar gene structure, 2 coding exons and a smaller 3' untranslated region. While less is known about GDF3, it's thought to be a key developmental regulator. And interestingly, multiple missense variants have been identified in patients with ocular as well as skeletal phenotypes, including Klippel-Feil syndrome. So we were really excited by these findings. In a smaller group of families, we've identified evidence for linkage to 2 regions of the genome, both of which happen to harbor genes that have been implicated in the autosomal dominant form of Klippel-Feil syndrome, which we know is associated with Chiari malformation. So our next step was to sequence these candidate genes within our families. So we've focused mainly on sequencing the coding regions of these genes. However, there are some predicted regulatory elements and non-coding regions of the genes that we've also sequenced. And the goal here is to identify a novel mutation or a rare variant that segregates with disease in families. So for GDF6, we're currently sequencing 96 affected individuals from 39 families. And for GDF3, we're sequencing 75 affected individuals from 28 families. And the way that these families were identified is we looked at our 66 families and chose those families that showed any evidence for linkage to either chromosome 8 or 12. So I only have preliminary sequencing results to share with you today, but what I'm showing here in this figure is we see the gene structure. Each arrow corresponds to a variant that was identified in our population of patients. Underneath the purple is the target region. So those are the regions that we have either already sequenced or are planning on sequencing. And then the green shows the uncovered regions. So you'll notice that there is a part of the coding region in exon 1 that we've yet to sequence. So that'll be important to look at. We've identified a couple of interesting variants so far. We've identified one interesting rare variant, and also we've actually identified one of the known Klippel-Feil syndrome mutations. So for the rare variant, we identified this in 4 Chiari patients from 2 families. And we actually see complete sharing of this variant within each of the families across affected family members. This particular variant occurs at a very low frequency in the general population. It's only found in about 1 out of every 200 individuals. And it's located within a potential regulatory region, so it might actually influence expression of this gene. So this is something that we definitely need to validate and follow up and look at potential functional effects of this particular SNP. The known Klippel-Feil syndrome mutation is a coding non-synonymous variant, meaning it's changing the amino acid code in the protein, so it's likely to have a potential functional effect at the protein level. This was identified in 3 Chiari patients from 2 families. This mutation has been associated with actually a wide range of phenotypes, so ocular, skeletal, as well as combined phenotypes. And there has been some functional work done on this mutation, and it's been associated with reduced GDF6 protein expression as well as reduced GDF-BMP signaling. So again, even with this variant, we need to validate it and continue to follow it up because we don't see sharing in one of our families across the affected family members. But that's something that we need to, as I said, continue to do some more sequencing and look at more closely. For GDF6, we've completed our initial passive sequencing, although there are individuals that have yet to be sequenced, so this is still preliminary. We've identified 1 known synonymous coding variant and 2 known non-synonymous coding variants. These variants occur at similar frequencies in our population as compared to the general population. In addition, sharing across affected family members was not observed in all cases. So at this point, we think these variants are unlikely to be involved in disease development, but our plan is to complete our sequencing coverage as well as look for potential regulatory elements and sequence those. So we looked at potential regulatory elements for GDF6, but we've yet to look at that for GDF3. So as I said earlier, we kind of took two main approaches, the qualitative approach, which I've just described, and a more quantitative approach in terms of identifying more homogeneous groups of families, and this was based on posterior fossa measurements. So here I'm showing in this figure the various posterior fossa measurements that we've been taking on sagittal T1 weighted MRI scans from both affected as well as unaffected family members when the data were available. Here's just a Pearson correlation matrix, and all this is showing is the pairwise correlations between all of the MRI measurements that we're looking at, and not surprisingly, many of these measurements are moderately to highly correlated with one another. So the first thing that we wanted to assess was heritability. So what proportion of the phenotypic variation is attributable to genetic variation? And so we estimated heritability for all of our MRI measurements using individuals from our 66 linkage families if the data were available. And we adjusted these for important covariates, including age at MRI, sex, ascertainment, measurer, as well as affection status. So here I'm showing the results, the heritability estimates for our single traits. If you focus on the top figure, I've highlighted all of those measurements that we found to be significantly heritable within our families. And what we find is actually a large portion of the posterior fossa does appear to be significantly influenced by genetic factors. And so this would be consistent with a genetic hypothesis for this disease. The table underneath just shows more detailed results. So for each MRI measurement, it's showing the heritability, whether or not that was significant, and then whether or not any of those covariates I mentioned were significant in our model. But rather than walking through this in any detail, I'd just like to point out a couple key findings. The first is our most significant finding, which was posterior fossa height, which was measured from the tip of the tentorium down perpendicular to the level of the foramen magnum. Not only was this the most significantly heritable trait, but was also one of the most significantly associated traits with affection status in our families. In addition, we found that the extent of tonsillar herniation did not appear to be heritable at all in our families. In fact, much of the variation in this trait appears to be more related to environmental factors as compared to genetic factors. So I think this is really something important for us to consider moving forward when designing genetic screens. You know, perhaps there are better ways for us to be classifying individuals as either affected versus unaffected. So at this point, we've examined a large number of MRI traits that are moderately to highly correlated with one another. So we wanted to take a step back and reduce the number of correlated traits we are examining. And basically identify a smaller number of variables where each variable reflects a more complex representation of the posterior fossa. So the approach that we're using to do this is to perform a principal components analysis, which can be used as a data reduction technique. So the input for this analysis are all of those individual MRI measurements, and the output will be the same number of principal components, where each principal component is a linear combination of weighted input variables. So we can use this to then identify a smaller number of principal components that account for the majority of the variation in our data set. So basically take a large number of variables, reduce it without losing that much information. We then use these to estimate heritability, still adjusting for those important covariates. So here I'm showing the 15 factors that came out of our analysis. The first several factors do account for the majority of the variation in our data. To give you just a little bit of a sense for what each factor represents, I'll show factor 1. So what we're looking at here on the x-axis are all of the individual MRI measurements that we're taking. On the y-axis is the weight. So what we see is that factor 1 is really reflecting a combination of all of those individual MRI measurements, but they contribute differently to each factor. So in this particular example, we see that the clivus contributes, one of the most highly contributors to factor 1. We also see the basion going to our reference line, and the reference line was drawn from the internal occipital protuberance to the dorsum sella, and then also the basal angle and Bugard's angle. So because we wanted to reduce the number of traits we were examining, we focused on our top 6 factors, which collectively explain 87% of the variance in our data. So one of the challenges that can happen when doing these types of analyses is interpretability of these factors. And so here what I'm showing is across all 6 factors, I've highlighted those specific measurements that are the main contributors to each factor. So what we see is that different regions of the posterior fossa really correspond to different factors. We then use these to estimate heritability. And here are the results. So not surprisingly, all of these factors were nominally significant in terms of being heritable within our families. And this makes sense because we know that each factor is really a combination of those individual MRI traits. And based on our initial analysis, we saw that much of the posterior fossa did appear to be heritable. Our most significant finding is shown here with factor 4. It was highly heritable, and it was also associated with affection status. And this is also consistent with that initial analysis because we see that the posterior fossa height is a main contributor to factor 4. So now that we've examined sort of taking different looks at the posterior fossa, both at individual traits as well as these more complex representations of the posterior fossa, we wanted to take this information and apply it to our linkage screen with hopefully reducing heterogeneity. And so the approach that we're using is to perform an ordered subset analysis, which I'll describe briefly in the next few slides. So imagine we have a population of Chiari families where each family is shown here as a rectangle. And we have 3 disease genes, gene 1, gene 2, and gene 3. So if we were to take this set of families and run our linkage analysis, we would have limited power to identify a disease gene due to the heterogeneity. So the goal of an ordered subset analysis is to identify an important trait-related covariate that we can use to identify an optimal subset of families that show maximal evidence for linkage. So basically, identify an unbiased way to pull out all the red families and then look for gene 1. So in this example, we'll use posterior fossa height, where each family is shown here 1 value. And that value is computed by taking the average posterior fossa height across the affected family members. The next step is to order our families by this covariate. And if we ran our linkage analysis at this point, as I said, we would find minimal evidence for linkage to any region of the genome. But using OSA, we can identify a subset of families defined by posterior fossa height that provide maximal evidence for linkage, and in this particular example, to the region of the genome that has gene 1. So we use this approach for Chiari. The candidate traits that we used, we basically focused on those posterior fossa traits that were found to be both heritable as well as significantly associated with affection status. And that left us with 10 candidate traits for the analysis. We had 6 of the individual MRI traits and then 4 of the factors that met those criteria. So here I'm showing our top 10 most significant results for the ordered subset analysis, and I'll kind of briefly walk you through the table. So on the left-hand side shows the chromosome for the result, the covariate, meaning the posterior fossa trait that was used to order families. Order is showing how those families were ordered using that covariate value, so either high to low, low to high, or optimal slice, which is really families are still ordered, but we use more of a sliding window approach. These 2 columns show evidence for linkage. So the unconditional LOD score is the LOD score obtained using all families. So this is really our baseline evidence for linkage. And what you can see is that we really have no evidence for linkage in any of these examples. whereas the maximum LOD score is a LOD score obtained using that optimal subset of families defined by the ordered subset analysis. And so you can see we're going from really no evidence to linkage to LOD scores exceeding 2 and even 3. The empirical p-value is just our way of assessing significance for our tests. The empirical p-value is determined by a permutation test where we basically permuted the family order, repeated our analyses multiple times to see how many times we're achieving maximum LOD scores greater than or equal to that result. And it's really asking the question, is there a relationship between that trait-related covariate and our evidence for linkage? And finally, this column shows you the number of families that were used in these subsets. So we're going from a large number of families to a much smaller number of families, but still seeing significant evidence for linkage. So we first focused on this region on chromosome 22, where we see with families that are defined by multiple covariates, we see increased evidence for linkage to this particular region. So I pulled this candidate interval on chromosome 22, and this is actually quite a large interval. There are multiple candidate genes, but one of particular interest is this gene EP300. This gene is actually very large. It's comprised of over 30 coding exons. It encodes a histone acetyltransferase, so it's thought to be involved in transcription via chromatin remodeling. It's also involved in chondrocyte differentiation, which is an important process for endochondral ossification, which is how we form some of the bones in our cranial base. And it's potentially involved in retinoic acid signaling. So this is particularly interesting because we know that pregnant hamsters that are fed vitamin A during gestation day 8 induces Chiari-like phenotypes in the offspring. And finally, EP300 mutations have been previously associated with the disease Rubinstein-Taybi syndrome, in a small proportion of cases, but about 3%. So interestingly, Rubinstein-Taybi syndrome has been associated with various craniocervical as well as spinal cord complications, including Chiari malformation, although this is very rare. As far as I know, there's only 3 reports in the literature. But in addition, other complications such as scoliosis, C1-C2 instability, vertebral fusion, large foramen magnum, and so on and so forth. So we were excited by these findings. Again, within sort of a smaller group of families, we see increased evidence for linkage to a region of the genome that contains both a biologically plausible candidate gene and a gene that's been previously associated with a disease that shares some similar clinical features with Chiari. So we wanted to look at what other candidate genes or what other genes were involved in this particular syndrome. So in addition to EP300, EP300 interacts with the protein CREB-BP. And mutations in CREB-BP have been identified in about 60 to 65% of Rubinstein-Taybi syndrome patients. So I've already told you how when we order our families by covariates such as posterior fossa height, we see increased evidence for linkage to the particular region on chromosome 22 that harbors EP300. So we wanted to see, well, in a similar subset of families, do we see any increased evidence for linkage to CREB-BP? So in fact, we do. It's not quite significant, but when you look at families that are defined in the same way, we go from no evidence to linkage on chromosome 16 to LOD scores almost approaching 2 in a very small subset of families. So this is sort of a similar story to what we saw with Klippel-Feil syndrome. In sort of a similar group of families, we're identifying linkage to 2 regions of the genome, both which harbor these genes that have been previously associated with the disease that shares features similar to Chiari. So now I'll end by discussing just a little bit of future work. We need to continue to follow up our linkage results, as well as complete our sequencing analysis of GDF3 and GDF6. We're also interested in looking at how copy number variation may play a role in disease development, so looking at particular regions of the genome that are either duplicated or deleted in individuals. And finally, we're conducting whole genome expression studies using patient tissue and blood samples. So the goal here is to identify more homogeneous groups of patients based on the gene expression profiles, and then correlate them to some clinical as well as radiological information. So there's a couple people to thank. I'd like to thank my advisors, Allison, Ashley-Koch, and Simon Gregory, who are both co-PIs on this grant. We also work with Dr. Enterline, who's a neuroradiologist at Duke, Dr. Fuchs and Grant, both are pediatric neurosurgeons at Duke. Heidi Cope and Jeff Steich are both involved in patient ascertainment for the project. Karen Soldano and Edgar Asimwe were helping with the GDF3 and GDF6 sequencing, and Caitlin Dunlap was an undergraduate researcher who helped with some of these MRI trait measurements. We'd also like to thank all families for participating in our studies. Without continued participation, we wouldn't really be able to do any of this research. And then, of course, from generous funding provided by several agencies over the years, including NINDS, ASAP, Conquer Chiari, and CSF. And would especially like to thank CSF for giving me the opportunity to come here today and providing me with travel support. And I'd like to take any questions. No questions? Yes?
That's just marvelous work, because, and it just shows how hard it is to just dissect out what a Chiari is and what are the things that make it develop, like a small posterior fossa, and you dissected all the elements, and it's just a miserable disease, really, for a geneticist.
Yeah, it's very difficult.
It's not brown eyes, that's for sure. And so, when you do all these comparisons, though, is that affecting your statistical power?
It is, definitely. So one thing that I didn't show, which I'm planning on doing, is for the stratified analysis that I was showing you earlier, I'm going to do a permutation test to assess the significance for that, which I haven't done yet. In terms of the ordered subset analyses that I showed you, those empirical p-values, those are not adjusted for multiple testing, but I can tell you that the top several actually do survive multiple testing correction, even when we account for independence across the test, which is actually pretty conservative.
Okay. And then the other thing is, I know the goal is always to get a lot score over 3. Well, I mean, obviously the goal is to find the gene.
Right.
Tell people where you are, but I mean, intermediate goal. But anyways, do you think that perhaps Chiari could be polygenic? In other words, you have multiple genes that contribute, like someone with diabetes or something like that?
Yeah, I think that's definitely possible. I mean, based on sort of what I was showing earlier, I think it's possible that there are multiple sort of major genetic effects, and there's likely a lot of additional modifiers to the disease. Because we do see in some families it does look fairly autosomal dominant in terms of mode of inheritance. I'm thinking it's not maybe as complex, but I don't know, and we won't know until we start to actually identify some more, identify genes and really start to dissect it. So I can only really guess at this point. But I do agree, I definitely think it is definitely more than one gene. Yes?
In some of our families, we've seen that there are more affected people than we would expect according to the Mendelian hypothesis, and I wonder whether you've seen that also and what your thinking is about that. I haven't looked in a lot of detail. I mean, we definitely see families that don't fit the autosomal dominant mode of inheritance. Whether or not that's due to that reason, or we see a lot of incomplete penetrance, where we're not able to figure out exactly what's going on. So it's hard to say. Again, I think it's more complex than just one gene going through. And I guess the goal of looking at these approaches is we're hoping that there's enough signal from some of these major factors to get that from these approaches, and then we can start to better kind of look at what additional complexities are involved in the disease. But I don't know. Yes?
So you're moving on down the line, you're 10 years later, 10 years ahead, and you've sorted this out, and you're ready to go to the environmental factors that may modify.
Mm-hmm.
How do people do that?
How do people do that?
How do y'all do that? I mean,
right.
Location, age, genetic background or ethnic background, or what is the...
Well, I mean, one thing that we've been trying to do is collect questionnaire data. And that questionnaire data, deals with looking at pregnancy history, what kind of vitamins were taken during pregnancy, whether or not birth traumas existed, illnesses or infections, and things like that. But the challenge, at least currently, is that we just don't have the data. We just don't have enough to kind of start to look at it. But that would be the thing to look at. So families that we have genotyped that also have this data available, then we can start to look at that aspect as well. Yes?
Are any of these loci have any repeat sequence abnormalities, so triplet repeats or anything like that?
Yeah, I mean, definitely. So actually, GDF6 has a repeat in the 3' UTR. So we're actually going to look at that as well to see if copy number of that particular repeat in the gene perhaps is correlated with affection status.
Because what Claire was saying about the increased frequency, I mean, you seem to see with the mothers that there's, through the maternal line, there seems to be an increased frequency. You see that in myotonic dystrophy, where it's a triplet repeat abnormality, and they have, the children born, if the mother transmits the disease, it's a worse disease than if the father transmits the disease, or more evident. I just wonder whether maybe, I don't know what that I never understood that mechanism. Maybe you could explain it to me, or is that something that could be going on here? Because when the mother is affected, it seems to really come down to the maternal line.
So I think, well, do you want to answer it? I'll give you the truth.
You can go ahead.
So what you were describing for myotonic dystrophy, that's genetic anticipation, and so it's more generational, really more so perhaps than gender differences. With Chiari, it's very interesting, and I probably don't have to tell this group this, but there do seem to be gender differences, which perhaps suggest hormonal influence.
Well, but with myotonic dystrophy, there is that anticipation, but there also is this maternal.
That's right, the
child that is gestated in a mother seems to have a worse disease than a child that is gestated in a normal mother.
Which again, perhaps is hormonal,
environmental,
or
could be epigenetic in some way.
Yeah.
So complicated.
It is.
So one of the things I've been wondering about, though, is whether or not there might be some mitochondrial contribution, because with the matrilineal inheritance that we do see in some of those families.
That's another good idea.
Especially because recently a lot of Chiari patients have also been found carriers of mitochondrial diseases.
Oh, really?
With what?
Mitochondrial diseases.
Oh.
Especially the more difficult ones. The guys that come with a lot of other baggage that if you weed out the Chiari, weed out the EDS, and many of them also have EDS, there is something you can count on. They started being tested and tested each other, the information from family to family, and many of them, much more than would be expected, just being a random association with mitochondrial disease.
That's interesting.
Now that they are classified in mitochondrial disorders, it's catching up. So maybe you can look into that as well.
Yeah, that's interesting.
So that's interesting in the sex issue because of the mitochondrial linkage.
Maternally inherited.
But the one thing I can say is that we have looked a little bit about inheritance coming from mom versus dad in our families, and we don't see an overwhelming amount of evidence saying that's one way or another. Well, I mean, it doesn't mean that in some families that might not be the case, but I can tell you in our 66 families we have sort of examined it, and it doesn't appear to be sort of an overriding theme of those 66 families. But yes?
First of all, congratulations. Really fascinating work.
Oh, thank you.
I'm very interested in your, and I like your principal component analysis approach to understand the mystery of the posterior cranial fossa. What is your interpretation? Is this because you are identifying regional markers, either localized regions that are more important, or is it because we are not considering, let's say, the third dimension and we are just subsampling the information? How do you interpret it interpret the regional aspect of your finding?
Right. So the one thing that I was actually a little surprised at when we did this, I mean, to be honest, I thought we might have some increased signal by looking more at regional components. But when you compare it to the individual trait analysis, we actually don't see as significant findings. In fact, when we examine it more basically, when we examine it at the individual level, we actually get stronger signals. So I'm not sure, but I mean, perhaps there are very specific regions as opposed to more of these modular units that is, at least in terms of our heritability analysis, seems to be more important.
Actually,
Does that make?
No, I would interpret what you found is that it's not localized, it's a global phenomena, and maybe every marker is not strongly correlated with the entire volume. And because we are not sampling the entire volume, you're kind of subsampling the space, and maybe depends how you sample it, that's how the strengths of your association. Would this be possible?
I guess it could be possible. So we haven't looked at volume. It's something that we've wanted to look at, but I'm sure, as you know, it's much more time consuming. So what we've wanted to do, at least partially, is look at the correlation between some of our measurements with volume in a subset of patients to see if what we're seeing is actually highly correlated with that as well, but that's something that I haven't looked at in any detail.
How about area? You looked at area in that single slice?
Yes, so I forgot to point that out, but in that single slice I showed a couple sort of, actually I can just pull it up, but we looked at posterior fossa area, and then we also looked at area in various components.
Yeah, it was one of the first slides.
This one right here.
Okay.
So you can see A1 through A5. So collectively, we're examining it, and then individually also.
So if you just look at the total area, is that predictive? I mean, is that by itself without doing all the other measurements?
Yes, it is, but not as much as. So posterior fossa area result is shown right there. So you can see it's heritable and it's associated with affection status, but it wasn't our most significant finding.
Measuring volume until a few years ago was time consuming because we were using cardiac method, etc.
Yeah.
Now with the mosaic system, it takes less than 5 minutes.
Oh, really? Wait, what system is this?
I'll show you later. It's
Okay.
available actually. He has it here. Show you.
Okay, great. Yes?
Christina, I know from Marcy Spear's paper in 06, American Journal of Medical Genetics.
Yep.
She thought there were maybe a correlation or linkage to chromosomes 9 and 15. And you mentioned 9 maybe, but where did 15 go?
That's a good question. So we don't see strong evidence on 15, and I haven't seen it in any of these subset analyses either. So, I mean, it's always possible with linkage to get false positives. It's also possible that, for whatever reason, this collection of families, we think it's genetically heterogeneous. So we may have just collected a lot more families that are involved with other regions. So it's possible later on, as we continue to ascertain families, it might pop up again, but we don't see it currently. Yes?
You didn't segregate the volumes above and below Twining's line, because in some of our measurements, you know, the clivus is shorter and the supraoccipit's shorter, and the volume beneath Twining's line seems to be small, but the volume above was normal.
Okay, yeah, so what I did is I looked at
A1, A2, A3 together.
Yeah, so I didn't do that. So that might be something to look at. So I looked at them individually and all combined, but I didn't separate it based on that line. But I can do that. That's a good suggestion.
Because the genetic development of the lower part of the skull is different from
Right.
from above, right?
Right, yep. So that's a good idea. Any other questions?
It was great.
Okay,
Thank
thanks.
you all.